yours.
Yeah, so thank you very much for, it's a great honor for me to be here and to present some of the work that the whole community in the world and our company as a part of the community is doing to enable next generation of mRNA-based therapeutics. So after a short disclaimer, let's go to the topic exactly. So this slide wasn't a member of this presentation like 5 years ago, because before that, mRNA therapeutics as a whole group of therapeutics was an experimental thing, and nobody really believes that in a few years we will see that in a therapeutic setting. But for now, we see that mRNA is actually the proven therapeutic modality with a global impact. Only in the first year, more than 7.5 million years has been estimated to be saved by only mRNA-based vaccines, which was produced by Pfizer and Moderna. So more than half of the people in the global population has been vaccinated by mRNA-based vaccines. So it's really the modality that actually saved a lot of lives during the COVID pandemic, and all of us have seen that. And it's not actually the great surprise that mRNA as a therapeutic modality comes first as a vaccine therapeutic modality, because both from the standpoints of the mechanism of action for mRNA therapeutics and also from the CMC perspective, so the perspective of chemistry, manufacturing, and control, mRNA is a great modality for the vaccines. I will not talk a lot about all of the reasons why that is the case, but the most important thing here is that mRNA is actually the truly digital therapeutics, and we create more and more strong understanding on the mechanism of design of such therapeutics, on the mechanism of action, with eventual beliefs that we will be able to design the exact vaccine that will work better for the pathogen, just from the structure and from the sequence standpoint. Another part is actually the ability to use the streamlined standard CMC process. So every type of mRNA actually manufactured by very similar conditions, manufactured with the similar quality controls, specifications, etc. So it allows us to use mRNA as a therapeutic modality for the pandemic situations, and it will have the shortest time to market as soon as we will need such kind of vaccine solutions. But why we don't see a lot of mRNA vaccines after this first approval, say 3 or even 4 years ago? It's because there is a long list of the shortcomings and problems with mRNA technology as a vaccine, as the same long as the list of advantages actually. And I will not go into each of one separately, but some of them are really connected to the nature of the expression platform, because mRNA is not best because it's on protein-only platform, and some of the pathogens that's required to represent the complex antigens like glycoproteins or lipopolysaccharides, etc., are not amenable to efficient mRNA therapeutic design, but we will talk about that later. Some of them are really connected to the efficiency of translation, because as we deliver mRNA that should be translated inside of the cells, you need to accumulate enough antigen, you need to present enough antigen to the antigen-presenting cells in order to initiate diverse and effective immune response that will generate the protective immunity. And it's not always the case, especially when we talk about the mucosal immunity, etc. And it's in part explained by the lack of expression efficiency for the most of pathogens, but also lack of the efficiency for the delivery system, because current state-of-the-art modality for mRNA therapeutics delivery, like lipid nanoparticles, are not very efficient in terms of the mucosal targeting. So where doctors see some roadblocks for the implementation, actually scientists see new research directions that just need to be tackled in order to enable further advancements of our mRNA as a vaccine modality. And currently, probably to summarize all of the directions the community works on, is actually focused in these 3 main directions. First of all, is actually the antigen design, because great vaccine starts from a great antigen that actually complies to all of the requirements that platform or pathogen gives in order to design this kind of antigen. Another part is the design of actually the expression system and the delivery solution for such type of expression system, either it's a viral vector or the mRNA or any other type. So for the sake of time, I will not talk about the delivery system because I'm also not the expert in this kind of things, but we'll talk a little bit about these two parts and some of the work direction that is right now actively pursued by the scientific community and by our company also. So let's start with some aspects of antigen design. I want to, if we talk about the utilization of mRNA as a vehicle and expression platform for vaccines, actually it's a quite complex in terms of the whole cycle that leads to antigen presentation and innate immunity induction. And mRNA as a platform not only pose some requirements on the antigen in terms of the antigen presentation, etc., but we also need to meet some of the requirements of the platform to efficiently express this antigen. So all of that complexity should be effectively tuned in order to obtain the effective vaccine. And there is a short, even very, I would say that it's too short list of some of the requirements that need to be met by antigen if you want to use mRNA as an expression platform for this antigen. But I want to stress out now that as for any antigen from any vaccine modality, the utmost importance is engineering of the synthetic antigen in order to maximize the presentation in the MHC of peptides that derive from this kind of antigens. And it's kind of quite a problem for a long time for the antigen designs for the vaccine solutions. But because what is actually influence the vaccine, the antigen presentation is a lot of factors. And when you talk about, and this, the main factor that actually influence this kind of presentation is captured in the structured antigen, in the relations between different parts of antigen, between sequence and processing systems of cells that will be behaved as antigen presenting cells. And if you start to talk something quite complex as features that need to be met in order to efficiently present antigen, the application of machine learning and in part some of the large language models is a great example of task when you can which can tackle such multidimensional space ranking. And it's actually the recent and most promising for now approach to predict and design the antigens in order to efficiently present it in MHC complex. When you're talking about the large language models and protein language models as a part of it, actually what we're talking about is mostly about embeddings in the matrix. So you do have some sequence of your antigens, example protein or peptide, etc., which is represented and encoded into the embeddings of such language models. Actually, you can see that as a matrix with each amino acid or codon or something else is represented by a vector, usually quite long in hundreds or thousands of parameters. And this representation is actually can be perceived as a coordinating multidimensional space for this such kind of antigen, peptides, etc. When we have such representations, which is called embeddings, you actually perform some of the operations in order to understand relations of these embeddings. And in language models, it's actually two kinds of operations. It's attention blocks and multi-layer perceptron blocks. It's actually the classical neural network workflow. By combination of such kind of blocks, your language model is actually learning some of these features inside this representation. So which amino acids is parity function, which of them are close in the three-dimensional space and influence on each other activity. Also, models actually learn a presentation of some physicochemical parameters of your antigens. And even the motifs that are imprinted into the antigens in order to efficiently bind different alleles of MHC. And all of this information can be learned even without labels. So from the structure of native antigens, you really can decipher a lot of this kind of features that will actually influence your MHC presentation efficiency. But it's not enough. And in order to efficiently predict the antigenicity and efficiency of antigen presentation, you actually need not only these structure derived features, but also the additional data that we currently have. So we do have right now corpus of immunopeptidomics data. So we actually know which peptides from experimental studies, from mass spectrometry studies, which peptides efficiently bind to different MHC alleles. We do know the data. We have hundreds of millions of eluted peptides from MHC complexes, from experimental antigens and natural antigens. And all of this info can be included into such representations in order to later rank some peptides on MHC binding and representation. Also, what is really important and cannot be learned from the proteins itself is the gene expression data. So we need to understand the levels of some MHC alleles expression in some cells and also the antigen expression per se. And we do have a vast amount of data of functional assays. So some of the peptide arrays, which are ranked in ELISpot, etc., can allow us to actually measure response of T cells from different origins to a specific antigen peptides. Integrating all of that, integrating all of that into such architecture allow us not only to include features from the MHC alleles and peptide alleles per se, but also add to this representation vector some of the features from functional studies. And having that, we really reach the uncomparable efficiency in terms of MHC prediction for the recent models. You actually see that the ROC curves show groundbreaking increase in performance while we incorporate all of the data into MHC binding prediction. Also, I want to talk a little bit about the expression system design itself, because it's another important part, as you already know. And when we're talking about the engineering of mRNA expression system, it's quite a multimodal optimization task, because a lot of components, a lot of structure elements of mRNA is actually influencing where, how, with which efficiency, with which kinetics the relative antigen and encoded antigen will be expressed. And today, usually how we tackle this multidimensional optimization problem, we try to bring them into pieces. Try to optimize different elements, and after that, combine all of the data on the high-level design of the mRNA template. And I want today to talk about the mental optimization of two main regions: 3' UTRs and ORF itself, because it's not as obvious what we can do in both of these directions. So let's start with 3' UTR. It's actually, it's only recently in last 15 years, and I would say up to we'll start to express synthetic mRNAs in cells, fully computationally designed. We'll see how important this 3' UTR in the activity and stability and protein output of mRNA vaccines. This is the data of our initial studies in our lab when we try to use to compare absolutely random certain nucleotide 3' UTR with what has been incorporated into the mRNA vaccines of Pfizer and Moderna during the COVID pandemic. And you can see that it's obviously that only change of this UTR region changed protein accumulation in multitude of times, reaching more than 8 times the total expression of protein just by changing of 3' UTR. Seeing that results, we actually committed to the screening of novel UTRs that was derived of cell-based libraries. What we actually did, we have our test cells. Actually, for our screening round, it was the cell line that was representative to our task. It was CHO cell line, but you can take any type of cell line you are interested to express your mRNA in. This alliance is actually incubated on the media with actinomycin D, and this media actually totally shuts the transcription of RNA polymerase II based transcripts. After some incubation, we harvest all the RNA, fragment it, and clone all of these fragments as 3' UTRs to our DNA plasmids that encode mRNA. So from that, we obtain already highly stabilizing fragments, which we survive with prolonged incubation in the media with actinomycin D. So it's actually the mRNA that can live longer in the cells. After the cloning, we actually produce the full mRNA